I'm Justin Wally. I'm at Iowa State University, and it's my pleasure to introduce our next speaker, Dr. Nikolai Slavov, who's faculty in the bioengineering department at Northeastern University, and also an Allen Distinguished Investigator with the Allen Frontiers Group, and director of the Single Cell Proteomics Center. And I guess as part of that, or spawning from that, he runs for the last several years, a nice summer school course on single cell proteomics. And if you're interested, you can find that online. I think it's in June this year. And if you can't attend, I know I've benefited from some nice YouTube videos that have been open and free to access. So with that, he's going to talk to us today about mapping the transcriptome and proteome of tissues in 3D at single cell resolution.
Thank you very much for this kind introduction, Justin, and thank you very much for inviting me to this symposium, which I have greatly enjoyed. I'm going to say from the beginning that we haven't worked with plants, even though, as you can tell from my background, I'm a lover of plants. But nonetheless, I hope that some of the ideas and methods that I'll share with you will be applied to mapping the atlas of various plants. The project in my group that is closest to building an atlas is one funded by the Chan Zuckerberg Initiative, and that is focused on building a three-dimensional atlas of both the proteome and the transcriptome and other omes, metabolome at single-cell resolution. And the approach that we take to performing this mapping is to use the high spatial resolution afforded by imaging of both RNAs and proteins. And based on imaging, we choose spatial markers that then we can use to reconstruct, to link to high-dimensional single-cell measurements of either transcriptomes or proteomes, so that we can computationally reconstruct the three-dimensional structures of both proteomes and transcriptomes with single-cell resolution. And while this project is going quite well, the results from it are mostly preliminary. So most of what I'm going to talk about today is going to be focused on the single cell protein analysis, which is a part of this last component. How do we measure the high dimensional single cell proteomes so then we can use to build the single cell resolution atlases? And as we think of atlases, it's useful to take a step back and look at what people have already achieved in building a joint RNA and protein atlases of different tissues and see what we learned from that. And one of the first thing to notice, one of the obvious results, is that when we localize messenger RNAs and proteins in human tissues in 2 or 3 dimensions and look at what are the various abundances, we see that the proteome maps of RNAs and proteins differ significantly. And this observation underscores the need to perform not just RNA-based atlases, but also protein atlases that can tell us more about the localization of proteins and the functions they perform in different parts of the tissue. And of course, to many of this audience, that observation will come as no surprise, because we know that many times messenger RNA abundances differ from protein abundances. Sometimes they differ because the data are very noisy. And indeed, when we see them differing, I think that's always a hypothesis that we should test. How much is measurement noise contributing to that? But in some cases of well-studied examples, such as P53, the tumor suppressor proteins, we know that much of this difference, that in this case I show here is observed across human patients, is due to most of the regulation occurring at the level of protein degradation. We know that P53 messenger RNA is constitutively expressed, the protein is more or less constitutively produced, and how much protein is present in the cell depends on how much is being degraded. It's normally actively degraded. And something interesting actually occurs if we try to do this kind of analysis of protein and RNA levels across different human tissues. we find that p53 is not such an exception, that there are many other proteins whose overall abundance can be predicted well based on the corresponding messenger RNA abundance, as shown by this correlated scatter. But if we look within a particular gene and compare the protein and the RNA level, they're less than perfectly correlated. And this is an example of Simpson's paradox. So it's quite important to distinguish what are we exactly comparing when we look at the RNA and the protein abundances for a particular gene, whether that's overall abundance or relative changes from one tissue to another. And then, of course, big part of the atlasing is to look at protein abundances, but as beautifully illustrated by the preceding talk, there is a lot more to protein function than just abundance. It depends what other proteins are bound or interacting with a particular protein of interest, what are their post-translational modifications, and so on. So it would be wonderful to be able to measure all of these things at single cell resolution and incorporate these aspects into the atlases that we build. And of course, there exist and have existed technologies for single cell protein analysis for decades that have mostly focused on using affinity-based reagents to detect proteins and have been incredibly useful. We've learned tremendous amount for variability of proteins across single cells based on these affinity-based reagents, based on fluorescent proteins. They have been instrumental for time-lapse imaging, tracing protein dynamics. But nonetheless, these methods offer relatively low throughput of analyzing maybe a few dozen, maybe 100 proteins per sample, but not scaling to a proteome. And oftentimes, their specificity can be challenging, especially in crowded intracellular environments. So we were interested when we started a few years ago working on this problem to push the sensitivity of mass spectrometry methods to be able to analyze single cells, together with other colleagues in the field, as Ryan Kelly beautifully illustrated their own path in pursuing this goal. So in deciding what path to take, for us, one observation that was instructive was the difficulty of coming up with a large number of standardized affinity reagents. This estimate published in Nature a few years ago indicates that annually we waste about $1 billion on purchasing affinity antibodies that do not have the specificity indicated on the label. And this waste is not accounting for follow-up experiments based on flawed hypotheses because of non-specific binding. And that was the bad news. The really good news that has always inspired me when doing single-cell protein analysis is how much more abundant proteins are than RNAs in a typical cell. On average, there are a thousandfold more proteins for every messenger RNA present in a cell. There are about 1,000 copies of the corresponding protein on average. And because of that, we speculated a while ago that we should be able not only to detect proteins, but also to quantify them. And there is a big distinction when we speak of single cell measurements between detecting and quantifying, because our methodologies rarely detect every single molecule in a cell. They detect a subset of the molecules, which is a sampling problem described by the Poisson distribution. It's like picking balls from a bucket. If we don't pick enough balls, we cannot possibly estimate the correct ratio between the different color balls present in the bucket, even if our measurement is absolutely perfect, even if we can ascertain without any ambiguity the color of the balls. If we don't sample enough, we cannot tell what the ratio is. And this has been really a major bottleneck for single-cell RNA sequencing methods to deliver quantitative information. So we speculated that while mass spectrometry might be less efficient in sampling proteins from single cells because we start from such a large number, we might be able to eventually sample more copies and be less limited by counting noise. I gather that most of the audience is well familiar with liquid chromatography tandem mass spectrometry. And with this slide, I just want to make the simple point that to determine the sequence of a peptide, we need to detect multiple fragments. It's not enough to simply see the precursor, which is relatively easy, and Ryan Kelly actually has published beautiful data from his group showing that even if we analyze a single cell, one cell at a time, we can detect tens of thousands of peptide-like precursors. But it's a different story to determine their sequence. To determine their sequence, we need to detect multiple fragments from each peptide so that we can tell what is the biological identity of the ion. And because some of these fragments are produced inefficiently, when we analyze single cell level abundance material, determining peptide sequences becomes very challenging. And this challenge motivated one of our first approaches to single cell protein analysis, which was the use of isobaric carriers. Uh, that's an approach that has been widely adopted by the community, and many of you are perhaps familiar with it. For those who are not, I'll just briefly review the core concept, which is that we can label peptides from small samples, such as individual cells, but we can use other small samples, so it can be biopsies or any other small sample of interest. We digest them to peptides, label the peptides with isobaric mass tags, and then in parallel, we perform exactly the same operation with a larger sample, which we call carrier. And then once labeled, the peptides are mixed together and analyzed by LC-MS. Upon fragmentation, of course, the reporter ions from the isobaric tags are released, which can be used for quantifying the abundance of the proteins across the different small samples, while the peptide fragments allow to determine the peptide sequence. And what is very useful in this context is that the peptide fragments come not only from the small samples, but some of them come from the carrier material, and they were, so since we selected the same peptide, they should be the same peptide fragments. And this allows us to detect more peptide fragments and enhances our ability to determine peptide sequences. Now, that's the theory behind it, at least. That is what inspired us to try it. But the question is, does it really work? And initially, when we started doing this, we looked at all sorts of correlations within the data and coefficients of variations. But ultimately, the most convincing evidence for me is to have an orthogonal measurement. And that was one of our priorities to do early on, to measure the abundance of proteins in single cells with a well-established method. And by this approach, by mass spectrometry, which we call SCOPE-MS. So in this case, I did not want to rely on affinity reagents. Rather, we expressed fluorescent proteins, mCherry and IRFP, in melanoma cell line. And then we measured the abundance of these proteins by fluorescence on the sorter. We measured their abundance by mass spectrometry, and the positive correlation seemed encouraging at the time. We've improved the accuracy of the method much since those early days, but I like keeping this slide for sentimental reasons. It's the first time that I knew there is orthogonal validation of what we were doing. And currently, we have built upon that idea and developed other supporting technology. As Ryan Kelly already explained, sample preparation is key to generating high quality data. And we have come with a couple of different generations of sample preparation. The first generation was to use focused acoustic sonication to avoid the use of chemicals that then have to be cleaned up from the samples. And that was low throughput and couldn't be automated easily. There was potential to do that, but it was going to be awkward. The second generation that we use quite a lot is a freeze heat cycle that we called MPOP, and we validated thoroughly to extract efficiently proteins for mass spectrometry analysis. There are many other simple methods that we tried that didn't work nearly as well. And then all of the sample preparation we currently perform in 384 well plates, automated by robots that can pipette into the nanoliter region. And then we perform LCMS analysis. We have tried using low flow and some of the top performing chromatographic columns and other approaches to enhance sensitivity. But for the most part, most of our data we generate by using relatively standard 75 micron inner diameter columns. And we prioritize robustness over squeezing the ultimate performance out of the system, while there is certainly a great value and virtue in also squeezing the ultimate performance. We just generally prefer to operate in the robust mode of generating large amounts of data. We like to use computational frameworks to optimize our parameters for data acquisition. We found those to be incredibly useful because before we started using them, I was spending a large fraction of my time analyzing failed experiments and trying to figure out what went wrong. And then we simply wrapped up all of the simple debugging capabilities that we were using into a data-driven optimization for mass spectrometry, Shiny app that is freely available for everybody to use. And it's useful not just for optimizing ultra-sensitive analysis, but can be used for any type of mass spectrometry optimization. At least we find it useful. And then we've used additional algorithms to help us enhance interpretation of the data. For example, we like to use retention time information to enhance confident peptide sequence assignment. We were very concerned of using matching approaches, matching ions that did not estimate false discovery rates. So we aim to develop a rigorous Bayesian framework that would estimate the probability of wrong sequence assignments. We also have a next generation method for doing the sample preparation, which is we're just about to upload on the bioRxiv as a preprint that uses Cell-in-1, which is a piezo dispensing commercial instrument that allows to dispense low picoliter droplets. And with that, we can, with this instrument, we can prepare single cells for mass spectrometry in volumes in the range of 4 to 15 nanoliters, and even low if we wanted to. We have found that pushing the limit below that, it doesn't seem to confer significant practical advantages, though it might for certain applications. And improved sample preparation clearly also improves the quality of the data, as in this case illustrated by principal component analysis of two cell types, monocytes and HeLa cells. And as you can see, the first principal component captures a vast amount of variance in the data. And this method we use not so much to distinguish different cell types just for cell clustering. We use much more to look for variability within a cell type. So with this technology now we are able to clearly identify both similarities and differences within the cell division cycles of those cells, for example. So we are the technology that we have is well past the point of just being used for classifying cells. We can look at much finer variation within a particular cell type. And I'll illustrate a little bit of our work and the capabilities that we have with this example where we focused on differences between macrophages that differentiate from clonal monocytic population. And the reason why we were inspired by this problem is the tremendous phenotypic plasticity of macrophages. They can vary from being pro-inflammatory and help to devour tumor cells and pathogens, or they can be anti-inflammatory and they can protect tumor cells from the immune system. So we wanted to see if some of that variability among macrophages will be observed even in the absence of external stimuli for their polarization, such as in the absence of cytokines or different extracellular environment. And to this end, we differentiated monocytic cell line into macrophages, and then we sampled both monocytes and macrophages at the end of this experiment for single cell protein analysis by mass spectrometry and for single cell RNA analysis by 10x Genomics, which is a popular commercial platform for single cell RNA sequencing. Um, Based on the data, we could easily do the most obvious thing, which is just separate the cells based on cell type. And what was encouraging in this case is not only that we could see the separation, but that separation accounted for a large fraction of the variance in the data. We can also see immediately that the monocytes are more form a tighter cluster as compared to the macrophages, which suggested that there might be more heterogeneity within the macrophages, but we couldn't confidently conclude this just from the PCA plot. So I'll show you a little bit more analysis to dig into that. And in this left PCA plot, we color-coded the cells simply based on knowing their identity. We kept track of it as we isolated them on the fact sorter, but we can also easily determine the identity of the cells simply by color-coding them with the mean abundance of marker proteins for either monocytes or macrophage cells, shown here to the right. Here on the left, we are showing some more validation that the proteins that we use to separate the monocytes from the macrophages have exactly the functions that we would expect them to have. In particular, the monocytes have higher abundance of proteins involved in proliferation, as one would expect for a cell line that divides, as opposed to macrophages that are adherent cells. And there we see enrichment for cell adhesion-related proteins and immune functions. The thing that wasn't expected and was more interesting in this data set is that we found a lot of heterogeneity within the macrophage cells that originated from this clonal population. And much of this heterogeneity correlated to this one axis of separation. So we were interested, what is that axis? What does it correspond to? And we looked at the proteins that are more abundant at either the macrophages on the left or the macrophages on the right. And we found that there is a perfect correlation of this axis of separation to previously identified markers for either the M1 pro-inflammatory or the M2 anti-inflammatory macrophages. And this result is indicating that we have this spontaneous polarization of macrophages in vitro, even in an environment that is as homogeneous as we could make it, and even without having provided any external cytokines for that polarization. Now, we speculated a while ago that we should be able to do more than detect proteins, but actually quantify them by sampling a reasonable number of copies. And we wanted to test whether this theoretical prediction expectation is borne out by the data. So we estimated the copy number of peptides and proteins that we detect per single cell per protein. And we found that on average, we sample over 20-fold more copies of peptides and proteins than we sample from messenger RNAs, as you can see here with the red distribution for the proteins compared to the blue for messenger RNAs. And what that's indicating is that in contrast to the very high counting noise associated with measuring RNAs by 10x genomics and most other methods, the measurements that we make by mass spectrometry of the proteins that we are able to quantify is relatively low, the counting noise. And this relatively good copy number of peptides and proteins being sampled corresponds to relative quantification in the single cells that is consistent with the quantification for relative protein abundances measured with more conventional bulk methods for these same cells. So since we collected joint protein and RNA data, we were interested in performing that joint analysis and seeing what we can learn from both data sets together, what are the similarities and the differences. And since I don't have much time to delve into detail, I'll just quickly summarize some highlights. We matched the two data sets by performing a correlation protein analysis, and we identified two clusters of genes, a large cluster where at least qualitatively the protein and the RNA levels showed similar trends, and a smaller cluster where the trends were opposite, and that smaller cluster was enriched for proteins involved in signaling activities such as RAP GTPase activity and others. We also performed joint dimensionality reduction using CONUS for the protein and for the RNA data. And we found that the clusters corresponding to monocytes and macrophages have the same centers, but the cluster formed by the proteins was much tighter, more coherent, while the cluster formed by the RNA cells was more diffuse, which probably reflects both higher technical variability in the measurement and high biological variability of RNAs across single cells. We did not see strong evidence for any batch effects that are uncorrected, since by color coding cells by biological replicate or technical replicate resulted in uniform distribution of these cells in the low dimensional projections. Now, one thing that I'm really very interested in doing, and we just started to move towards, is to use this variability observed between single cells to infer potential regulatory mechanisms. And that's a very old idea, that the regulatory interactions should be reflected in the observed joint distributions of proteins and RNAs, but it has been fairly difficult to obtain data to perform this analysis. So with the data that we already had, we tried to do it, to move in that direction. And we ordered the cells by using common principal component analysis. So here I'm going to exemplify the results with a couple of relatively representative genes. So here is a representative gene from the cluster 1, where protein and RNA levels showed similar trends, and we see this fairly strongly correlated joint distribution for a highly abundant protein. Now, if we look at the less abundant protein from cluster 1, we see that again the joint distribution is correlated, but in this case, we begin to see the discreteness in the RNA data, both here in the, as we have ordered the cells based on the RNA levels, and in the joint distributions, which corresponds to having counted 1, 2, 3, and so on messenger RNAs. And that's not present at the protein data because we sample more copies in part. Now, it was particularly interesting to look at p53, which is a protein that shows very little correlation, if any, between the RNA and the protein level. And then we could go one step further, and we could ask whether the transcription factor p53 can predict the abundances of its target messenger RNAs, either those that are activated by p53 or those that are repressed. And what we found is that the p53 messenger RNA is not informative, cannot predict the activity of the transcription factor, while the protein abundance of p53 was informative, at least for the abundance of the repressed targets of the transcription factor. The protein abundance wasn't informative for the abundance of the activated targets, which most likely reflects the fact that we don't account for protein localization to the nucleus, post-translational modifications, other transcription factors that interact together. But nonetheless, I think it's encouraging to see that protein levels are already proving to be more useful in determining regulatory interactions and messenger RNAs of the levels for the transcription factors. And with this, I would like to thank my team that has made everything that they shared possible. It has been the result of a very collaborative effort among many students in the group, and in particular effort led by Harrison Speck. And I'll stop here by acknowledging the funders that have made it possible and inviting questions.
Thank you for that wonderful talk, and got a couple questions in the chat. First off, could you comment, this is from Sesway Chen, could you comment on the booster channel contamination issue with MS2, and should we be using MS3 for quantification?
Well, so this is generally when we use isobaric labeling, whether we use isobaric carrier, that is something that some people call booster, or not, there is a potential of co-isolation that, of course, affects quantification. And one of the strategies, certainly not the only one, but one of the strategies that is popular to reduce that co-isolation is to use MS3, which of course comes at the expense of reduced sensitivity. I think it's a reasonable option to use MS3 to try to reduce co-isolation. But there is a trade-off there, which is you will reduce the sensitivity in the copy number of peptides detected. One particular aspect that I think has been very helpful for us so far is generally speaking, co-isolation is much less problematic for the most abundant proteins than it is for less abundant proteins in the proteome. And to the extent that our coverage has been relatively shallow, we have looked at 3,000 proteins across many cells. In any one cell, we usually quantify only about 1,000, but our entire data set analyzed about 3,000 proteins. They're among the most abundant proteins that are least affected by co-isolation. So I think that by using MS3 approaches, one might actually incur larger errors on the side of sampling too few copies from these slowly abundant single cell proteins than one might gain by reducing the degree of co-isolation.
Fascinating. I think now for time, we'll move on to the next speaker, and then I'll look forward to seeing you again in the panel discussion.